partner here at DGU, and Ramon is going to tell us about revolutions in gene regulation, paradigm shifts, or conservative transformations. Ramon, please.
Thank you. Thank you for the introduction, Phil, and thank you for inviting me here. My lab is working on gene regulation, basically trying to understand how mutation in gene regulation cause human disease. So mainly we'll be focused on the human genome. Okay, so there was a great introduction here by the previous speakers. So I will just continue with Kuhn's central concept that paradigm shift, what is paradigm shift, and if it's part of a revolution or not revolution. So my point of view is the paradigm shift, meaning that first you have paradigm, meaning you have concept, you have some terms in a specific field, and try to simplify something and make it with common terms, common jargon, so all the scientists in this field can talk. And then there is some exceptional or things that crack in the paradigms that then at some point someone finds this crack, and I think this is part of paradigm shift. So today I will focus on three things. One is coding and non-coding sequences, then about transcription factors, and enhancer-promoter interactions. So a brief overview about the timeline of the human genome. So we start with genetics or Mendel law, and then 1952, the cracking of the DNA structure by Rosalind Franklin, and of course Watson and Crick, which make a real breakthrough, revolution, depending who we asking now after Rosenberg talk. And then in 1961, it's the finding the genetic code. And then what really make us to sequence the human genome is the two technologies. One is the Sanger sequencing at 1977, and the other thing is the polymerase chain reaction, PCR. So by those two that we can amplify sequences from the genome, and we can sequence them. This is basically what led, these two technologies led us to the ability to sequence the human genome. So maybe it's difficult to imagine these days that before the 2000s, we didn't have the reference for the human genome sequence. And so with these two technologies, the Sanger sequencing and the PCR, we were able to start sequence genes and find mutation, as written here, the cystic fibrosis, BRCA1. And then in 1990, they start the Human Genome Project, meaning they divided the genome built from chromosomes in different labs, sequence different chromosomes, and eventually they start sequence by Sanger and PCR, each group different chromosomes, and together as a consortium they trying to complete this task and to sequence the entire genome. So in the meantime, till they complete that, different genome was available, was sequenced as the influenza because it's small, okay, and then 99, chromosome 22, was completed. Again, Y chromosome 22 because it's the smallest one, so it was quicker to sequence it, and then after that is the Drosophila and the mouse genome, and eventually the first version of the human genome was at 2001, but completing or announcing the completion of the Human Genome Project was 2003. What this gave us? So, the Human Genome Project allow us to have the reference sequence, and then they discovered the main discovery. Many discoveries from that, but the main discover is that there is about 20-25 genes in our genome, and the genome contains 3 billion base pair, and those 25,000 genes are basically less than 2% of the human sequence. Meaning that, as you probably most of you know, what's called the junk DNA. At the beginning they thought that these sequences, non-coding sequences, are meaningless, and they call it junk DNA. So we know, today, we know that this is not true. And in these non-coding sequences, there are many, many regulatory elements, as you see here. It could be promoter, enhancer, silencer, repressor, and insulators, and etc. And this is only part of the regulatory elements that exist, because there are also others like microRNA, non-coding RNA, etc. We will focus on, basically in this talk, on enhancers. So, what this gave us is starting to ask the question, what is this large amount of DNA that does not code for protein? So, if we look now on the sequencing cost, I guess most of you are familiar with this diagram. We know that at the beginning, when they complete the Human Genome Project, the cost, they said it was $100 million. Eventually it was much more. It was 2.7 billion. And then with time, the technology of sequencing was really improved, and the cost was lower and lower. And I think one of the breakthrough was around 2008, when what's called the next generation sequencing, or massively parallel sequencing, this technology came out and really improved our ability to sequence DNA sequences. And eventually today we are standing that of the cost of sequence the entire human genome. It's between $200 to $600, and there are different companies that claim and show some technology that they will able to reduce us to 100 and even $18 per genome. So this is, I think, from 2008. This is a really revolution in our ability, a technology revolution. That the impact of this technology I will present now. So, as you probably familiar, there are in the version of the human genome, there was gaps of region that we couldn't map in the genome, mainly because there was repeats and with the short reads, with the technology of massively parallel sequencing, it was difficult to sequence. And then it took another 20 years, and eventually in 2022, they announced about the telomere project, when they complete to sequence the entire genome. And again, this is basically was due to the technology of long reads. Instead of sequence only a region of element of a couple of hundred of base pair, we were able to sequence with the technology of nanopore or PacBio. We were able to sequence much more longer read, which able to go over this obstacle of repeats elements as in telomere and centromeres and region with duplication and regions that was difficult to sequence. So back to the genome. So after in 2003, after they complete the Human Genome Project, there was another consortium, what's called the Encyclopedia of DNA Element, the ENCODE project, where this consortium was focused on understanding the non-coding, not only, but mainly the non-coding sequences in the genome, and it was completed 2012. And basically, the announcement is that more than 80% of the human genome can be assigned to a specific biological function. And basically, there are more than 4 million regulatory elements in the genome. What does it mean? So gene regulation can affect on gene expression, and gene expression is involved in many different areas: development and differentiation, morphological changes, human variation, pharmacogenomics and human disease, congenital disease, or other like cancer, things like that. So what is the revolution here, or I don't know if revolution, the paradigm shift is that these non-coding sequences are actually have some regulatory effects, meaning that they can control the activity of the 25,000 genes in different ways. And based on the ability to sequence this massively parallel sequence technology, it's improved not only the ability to and reduce the cost of sequencing of the human genome, but also developing different techniques, as it's written here, if it's ChIP-seq, RNA-seq, chromatin interactions, such as high-C, 5C, we'll talk about that, that allow us to really starting understand what is behind these non-coding sequences. So if we look on bacteria compared to eukaryotes, we can see that the regulation of transcription, and again, I'm mainly focusing on transcription regulation. And you can see so we can see that there is the promoter region. Okay, this is the transcription start site, where the transcription starts, and from here there is the RNA. The RNA will transcribe and then translated to protein. And there are different regulatory elements, okay, like enhancers, distal enhancer, insulator, silencer, and even enhancers that are downstream to the gene. So this is much more complicated. And basically what the paradigm was, or the concept was, that there are protein coding sequences, which are the genes, and there are non-coding sequences that basically contain the regulatory elements that control the expression of the genes. So what we were interested, because we were interested in human genetics, we look for if coding sequences can also function as enhancers. Okay? Meaning if DNA sequence that codes for protein, if it can have also another function. In our case, we look on enhancer. I will show other example. So what we look here, this is Naama Hirsch work. We look on genes here. There is gene A, okay, that deletion or duplication or inversion or translocation in that gene can lead to phenotype A due to disruption of the coding sequence of gene A. However, it could be also that there are regulatory elements such as enhancers that control the expression of the nearby gene, gene B. So actually, because we delete here this exon, okay, and this exon also what it's doing is function as enhancer, meaning it's recruiting some transcription factor and is doing this looping and activate or induce the RNA polymerase to start the transcription of gene B. So basically, if this is the enhancer that controls the expression of gene B, its deletion can lead to phenotype B. So now we'll show you an example for that. Okay. So we have two genes. It's HDAC9 and twist1. And mutation in twist1, in the coding sequence of twist1, of this gene, called craniosynostosis, meaning it's pre-fusion of the suture of the skull. And then what happened is that the skull cannot grow after birth, and our skull is growing till age of 24. So it cannot grow, and then it leads to different problem or in the skull or even neurological problem. What I want to show you here is that we found, okay, each here bar, the red bar, showing deletion in patient with craniosynostosis. And what you can see, what I will mention here, that all this deletion in red are covering or deleting the HDAC9, the nearby gene for twist1. So it's not affecting the coding sequence of twist1. However, what they have craniosynostosis, meaning that some of the regulatory elements that control twist1 are located here in the coding sequence of HDAC9. And when we model this phenomenon in mice, so mutation of twist1, the heterozygous mutation, lead to similar craniosynostosis phenotype, and also to polydactyly, to extra finger, which is another phenotype of coming with craniosynostosis in the patient. And what you can see here, that when we knock out one of the enhancers of that control twist1, that also function as HDAC exon, it's leading to similar phenotype, craniosynostosis-like phenotype and polydactyly. So by that, just to show you that this paradigm, that there is coding and non-coding, and the non-coding regulate the expression of the coding sequence. So here we show you one example that also coding sequence can function as a regulatory element. And we are not the only one that show it, of course. Okay, there is another paper that came out after our paper, and what they show that they call it duons, meaning DNA sequences that code for protein, and they can also affect the transcription factor binding. These sequences are affected by the transcription factor binding to these sequences, meaning, they show other amplification of that it basically can control. Coding sequence can also have additional function. So this paradigm that coding and non-coding, again, I don't think it's a revolution, it's just a kind of a changing in the paradigm of the DNA sequences, and we show that even coding sequences can regulate the transcription of other genes. Okay, I will move on. So if we looking on bacteria, or what was the breakthrough or kind of paradigm shift in gene regulation, one of the very famous finding is the lac operon that Jacob and Monod show that different genes and a repressor of a gene of one gene can affect the transcription of another gene, and by that they can control the activity of the bacteria. Of course, in human, it's in the human genome, because it's a larger, it's a multicellular organism, and the amount of non-coding sequences encoding that regulating, or the percentage of the regulatory element in the genome, it's much higher than in bacteria. And there's more genes, and the genome is bigger, so it's much more complicated. And what I want to show, that it's not only like promoter based as in bacteria, it's much more complicated, as I showed you. And in that, we need to have a better systematic look on that, and as Ute already mentioned and present, it's Eric Davidson work about gene regulatory network, and he was one of the first that using Serogen to show how complicated all the gene regulation that is involved in early steps of development. So now we go deep into what is the levels of regulation in the human genome. And I just want to emphasize here a couple of things that hopefully it will not be too much information for those who are not familiar with what I'm showing here. So again, for transcription to occur, basically we need a promoter, we need RNA polymerase, okay, that will be based on the DNA sequence, will transcribe the RNAs that will be translated later on to protein. So to this for occur, we need, so promoter can actually activate the transcription by itself, but if we're looking for in development, so enhancers and other regulatory elements, they have a very important role in the spatiotemporal expression of genes. So this is what you see here. So for that to happen, the enhancer, which is a DNA sequence that binds transcription factor and associated the RNA polymerase to start the transcription, it's required a couple of elements, okay? One is that the chromatin will be open, and for the chromatin to be open, we need the histones that build the chromatin to be with certain modification that help to remove the nucleosome of the histone, so transcription factor can bind to it, okay? So we have one, we need that the DNA sequence contain binding site, we need the chromatin to be open, and we need then the transcription factor that will bind to the enhancer, and eventually we need that a looping will occur and the enhancer that can be located in different places in the genome will be in close proximity to the RNA polymerase to activate transcription. Okay, so it's different level. How much time do we have? 10 minutes?
15,
15 minutes. Okay.
including discussion.
All right. So if we are breaking this to different steps, okay, and we're focusing now only on transcription initiation and elongation. So again, the DNA sequence contain the template and the motif that transcription factor will recognize and can bind to. Okay, we need the transcription factor to be expressed in this specific cell that they are required to be controlled or to express that gene. And then on the epigenetic, we need the chromatin to be open. One of the things that the chromatin should, so it will be open, it requires some epigenetic modification. And lastly is how the chromatin is organized and the high order organization of the chromatin, and that the DNA, the enhancer, can do the looping as showing here. Okay, so I hope it's not too much details, but now I want to show you a couple of, after this was discovered that all these elements are important for the spatiotemporal activity of enhancers that will regulate the expression in specific cells, specific time, and specific level for, let's say, for development, for normal development. So we will focus on the enhancer and how it looks like its work. So if we have the promoter here and the exons that eventually will be transcribed and spliced and get generate the RNA that will be translated to protein. So here you see in situ hybridization of where this gene is expressed, and basically there are 3 different enhancers, okay, that control the expression of this gene in each of these tissues. Okay, so there is one element enhancer, okay, that control the expression of this gene in the limb, one in the neural tube, and one in the brain. So all together, okay, is give the expression of that gene in this mouse embryo. Now, if we look on this, what is required? If, for instance, one of the enhancers located in the intron of the intron 1 of this gene, what it means? It means that this enhancer should activate the RNA polymerase that's sitting on the promoter, and then this enhancer needs also to be transcribed. Okay? So how does it work? How it could be that this DNA, okay, first it's activate the transcription, and then it's itself, it's an intron, it's not an exon in this case, but it's required to be transcribed. So there is like, I don't know if it's clashing here or not, but this is part of the things that we still don't understand how exactly it's work, Okay, and this is kind of the challenges that we have in the field. We have another issue, okay, which it's redundancy. Okay, so we have could be two different sequences that basically activate the transcription in the same tissue. Okay, and when we look on this element, their sequence is different. They don't have the same sequence. They don't have the same motif, but they actually control the expression of that gene. And if we delete one of the elements, or it will be mutated, the other sometimes can compensate on the lack of the mutation of this one. So how exactly it works is another question that we don't know how to answer. And a third issue with transcription factors and enhancer, it's what we call pleiotropic enhancer. What is pleiotropic enhancer? So I will show you here. So this enhancer, okay, activate the promoter and the RNA polymerase on the promoter and leading to transcription. And you can see here in this zebrafish model, okay, when we took this enhancer and we clone it into a GFP green fluorescent protein as a marker. So you can see that at early development, this enhancer drove this gene and it's expressed in what we call the branchial arch. But later, when we look on through development, it fade out, it stopped function here, and it start to be active in the notochord. So we have the same sequence that can activate transcription in one tissue, and later on, it's becoming active in a totally different tissue. How that is work, okay? We still don't know. This is what we say. What is I want to mention here is this is based on the transcription factor, and the current model is the billboard enhancer model. So we have, for instance, here we have enhancer in different cell type, and it could be that different transcription factor can activate this enhancer. It could be that there are some transcription factors that are common that need to be bind to this enhancer in all tissues, all cell type, to activate this enhancer, and other more specific, like here the green one, is more specific only to certain cell type. So with that, the question is if this transcription factor are leading player in gene regulation or not, and probably yes, but definitely they are not the only one. And this is kind of the paradigm that this is the current paradigm, but still there is some many example that this model is not fit to what we observed. And last thing that I will do very short, I will talk about very short because I ran out of time. So it's not about, when we talk about enhancers and transcription, it's not only about the binding of transcription factors to the enhancer, but also about this enhancer need to be in close proximity to the promoter to activate the RNA polymerase to start transcription. So if we have different enhancer in each tissue, we need different chromatin organization. So one of the technology that made, let's say, a paradigm shift or really help us to understand how chromatin looping are affect gene regulation is all this kind of chromatin conformation capture and other methods that are mainly based on massively parallel sequencing. I just will present here the IC and the ideas that I will not go over it, but just this is a method that allow us to find how the chromatin is organized in the cell, okay? Which element or interact with which element in the genome. And with that, what these all these methods of that based on chromatin conformation capture or HiC, they define what we call today topologically associating domains, which are certain regions that of the chromatin, okay, that likely to interact with each other. Okay, let's do it as simple as possible. So what, if you look here on this part of the figure, and I will not go into the detail, I will explain it very briefly and simply. Okay, so we found, if we look on the chromatin, there are regions that are likely to interact with each other more than their neighbor sequences, and they could have different functions. So if they are really condensed, all the gene in that region will be silenced. Other region could be more open, still interact with each other, but more open, and over there gene can be active, okay, or repressed, or regions that are more like nothing happened over there. They're just kind of there's no too many genes over there. It's not active, okay. So basically, what I want to say that if we look on two different cells and they have two different functions, meaning they have different genes that need to be expressed and regulated. So with that, we thought that the chromatin organization in those two cells will be totally different, so different enhancer can activate different gene. But basically what was found, okay, that the structure, this is what you see here in the triangle, the red triangle, the structure of how the chromatin is organized between different species and different cell type is basically the same, okay? And this was a really change in our understanding how the chromatin is organized, and without of lack of time, we'll just go to the end. I just want to mention here Stavros' work about interchromosomal interaction, but I will let him talk about this tomorrow. And I will finish with that, okay, that in gene regulation, I don't know if, as Ellen mentioned, I don't know if it's a revolution, paradigm shift, or only development of technology and our knowledge, but what I try to show you today is one that the dividing coding and non-coding regulate the coding sequence. It's not always correct, and transcription factors are definitely a main player in transcription regulation. However, they are not the only player. And the last thing is enhancer-promoter interaction. We thought, okay, that it's really required that promoter enhancer will interact to act, so the enhancer can activate the RNA polymerase on the promoter. But Stavros and other lab like Wendy Bickmore show that it's not always the case, and there's some cases that the enhancer and the promoter can fluctuate and can be in a larger distance that people find till now. With that, thank you very much for listening.
Thank you, Ramon. One quick question. Yes, please.
Thank you for your talk. I'm from physics, so I don't know much about the chromatin, but what I do know is that it's pretty closely packed and complex. So when you said that people look at interacting regions, how do they exactly define the regions? Because there are like, I guess, millions of regions, if I understood correctly.
Yeah, so basically the common system or method is by sequencing, okay? So you find some regions that interact, or sequences that interact, you sequence them, you know that they are in the same complex, you sequence them, and then you align them back to the genome, and you know, okay, this sequence came from here, this sequence came from that, okay, and then you know that these two regions interact with each other. Did I answer your question?
Yes. Thank you.
So we have time for another question until we activate the presentation of Anna. Okay, thank you, Ramon.